Okay, good night everyone, thanks for coming. From ECUSA, California, we want to thank to each of you for being here with us today. We are really pleased to be able to welcome all of you, because this is our first face-to-face event since the pandemic started. Then it's very, very special for us. Thanks again for coming. First of all, big thanks to our sponsors, the Embassy of Spain and Spain Art of Culture and the Fundación Ramón Areces for the support, because otherwise we would not be able to be here. And of course, thank you to ECUSA. I will thanks especially to our board of directors, Judith and David, that help us a lot for this event. And I would like to start welcoming our guest. Welcome, Dr. Eva Nogales. Thank you very much. She's our keynote speaker today and also participant in the round table. I have to say that even if you don't know, you motivate all of us, the organizers, to move this event forward, because you were really available from the very first minute, and thank you very much. Welcome to our other roundtable participants, Juan Lama, Raúl García, and Ernesto Criado. We will introduce them later on. And I would like also to welcome Doina. She's a representative of the Embassy of Spain. She's flying from DC. Then I would like to welcome her as well. We are really honored to have all of you today. Thank you. Next, I would like, of course, to introduce our ECUSA organizers. I don't know if you want to come here, because it seems easy, but this has a lot of work. Then they deserve a little thank you as well. Irache and Timo are our newest volunteers. Thank you so much. They have a lot of good energy and good ideas, and we are really happy that they join us. Alba González, that is our more experienced volunteer in ECUSA, California, the oldest, not the oldest in age, but the one who is like, no. But like, we need her, then like, thank you very much, Alba. And Antonio, he's our vice president, and he has a lot of energy, he's very positive, and he's always willing to do projects, then it's like great to work with him. Thank you. I'm Gloria Hernandez. I'm the president of ECUSA California, the chapter of California, and I'm with them for like 3 years now, and I'm really, really proud that we are all here to do this. Thank you very much. Then our goal with this meeting, it was just to reflect what ECUSA mission is, that is to outreach science and also to promote the Spanish scientists, that like we are good scientists, and we want to promote that as well. But like during these kind of events, that they are like educational events, but also a priority for us is to create a good ambience and of networking and also connection between us. Then I hope that we will get it. That is at the end with the Spanish food and the drinks where usually we get it. Then, okay, with that I'm going to do a short presentation of ECUSA, and then we will start with the program of the event. Thank you. Okay, then ECUSA, as you know, we are an association of Spanish scientists. It's a non-profit organization, and we are, the people who form part of the organization are professionals of science and technology with an affinity of Spain. Not only Spanish people, but people that has an affinity with Spain. It was created in 2014 and is run exclusively by volunteers. We have 6 chapters: Boston, California, Midwest, New York, DC, and Texas. We are currently around 1,000 members, and from those we are 64 people who are like active volunteers, people who coordinate, do the events, and everything. As I say, they are like scientists in different areas. As you see, more or less equal number of women and men. Most of our members are postdocs, but there are also people who work in other areas, including assistant professors and people who work in the industry. And the distribution, you can tell that in New York is where there are more members. Then I invite you to join California because we are losing 11%. Then we need you to move this number a little bit up. Sorry. Then our missions and objectives, we aim to promote science, technology, research and development, and how we want to do that by organizing, as I said before, outreach and educational events like this one that involve the communication between the scientists. We want to establish a network of Spanish scientists in the US to facilitate integration of the newcomers and also to exchange ideas and experience. And we have a role as well in the science diplomacy. We are kind of like the bridge between the Spanish scientists and the American and Spanish associations, agencies. In particular, of like how we kind of have a more presential role is that now therefore we form part of the FECYT. FECYT is like an association of associations of the Spanish scientists. RISES represents 18 associations of around 4,000 scientists that they are working or studying abroad. Then the same, the role is to promote science, but the main function is to establish the communication with the Spanish Association of Science, Technology and Innovation, Agency for Spain. Then how we do that? We do that like through our programs, through our commissions. We create different workshops, training. We try to promote the scientists, and then we do like interviews or like small videos to kind of give the possibility to the scientists to show their research and their work. Then we have 4 main areas of work: integration, professional development, spreading and networking. And how we do that is through our 4 commissions, that is ECUSA, CDA, ECO and communication. I'm not going to explain anymore because I prefer to, like the theme of today is believing, right? It's better one email than many of my works. Then I'm going to play a short video, 4 minutes video of ECUSA, where people, the volunteers, not only in California, if not all parts, are going to introduce ECUSA.
En el año 2014, un grupo de investigadores españoles inmigrantes en Estados Unidos decidió promover y dar visibilidad a su ciencia. Con esfuerzo, paciencia e ilusión, se fundó ECUSA, Españoles Científicos en Estados Unidos. Con el paso de los años, el número de miembros ha ido creciendo de 5 a más de 1000, dando lugar a una creciente red de contactos que ofrece oportunidades profesionales entre sus integrantes.
Somos una comunidad de gente que nos apoyamos y nos ayudamos los unos a los otros.
Y tener contacto con gente que se produce una asociación, problemas.
Un pequeño trocito de casa fuera de casa.
A su vez, ECUSA favorece la movilidad de los investigadores, su integración en las comunidades locales y su posible regreso a España.
Me gustaría seguirla en España o en Europa.
Por todo ello, y gracias a su creatividad, ECUSA se ha convertido en algo más que una simple organización científica y divulgativa sin ánimo de lucro.
Aprender, compartir.
A día de hoy, ECUSA es comunidad, apoyo, ciencia y conocimiento, divulgación, oportunidad, aprendizaje.
Ha sido muy enriquecedor y muy importante a nivel personal.
A mí me ha dado la oportunidad de conocer a gente que sin ECUSA no hubiese conocido.
Y un sentimiento de sentirme útil en la sociedad.
ECUSA se organiza en 6 capítulos: Boston, California, Midwest, Nueva York, Texas y Washington DC. Y trabaja en 4 comisiones y 7 programas. Las comisiones cuentan con Mujeres en ECUSA o MECUSA, Desarrollo Profesional y Asesoría o Career Development and Advisory o CDA, Comunicación y Education and Public Outreach o ECUSA Educa EPO.
Y también destacaría que ECUSA es una máquina de hacer proyectos increíbles y de interés científico y para los científicos y la sociedad en general.
El objetivo principal de ECUSA es abogar por la igualdad de género en la ciencia, dentro y fuera de ECUSA. La CDA, o Comisión de Career Development and Advisory, tiene la misión de fortalecer, enriquecer y promocionar las carreras profesionales de los investigadores de ECUSA, poniendo el acento en las nuevas generaciones. Comunicación, por su parte, trabaja en la difusión de todas sus actividades y eventos. Esta comisión ejerce un papel fundamental como plataforma, informando sobre el trabajo científico realizado por los miembros de ECUSA y de muchos otros científicos españoles en Estados Unidos. La Comisión de Education and Public Outreach, EPO, a través de sus eventos y conferencias, busca aumentar la percepción social de la ciencia y la tecnología. Para ello, ha creado, por ejemplo, un programa dirigido a escuelas, con el objetivo de despertar la curiosidad y fomentar el aprendizaje de niños y jóvenes en el campo de la ciencia, tecnología, ingeniería y matemáticas, participando en ferias científicas y organizando talleres en castellano e inglés en centros educativos. En su estructura organizativa, ECUSA cuenta con más de 500 miembros, con el 67% de mujeres y 33% de hombres. El 15% de los miembros forman parte activa en la dirección, diseño, tesorería informática y asesoría legal de ECUSA. ECUSA supera los 5.000 seguidores entre Facebook, Twitter y LinkedIn, y con sus más de 60 eventos anuales, la gran comunidad de ECUSA se pone al servicio de la promoción y visibilidad de la ciencia, la educación y la formación de las nuevas generaciones de científicos, con el sueño de llegar a convertirse algún día en asociación de referencia mundial, y en la que el nombre de la ciencia e innovación se elevan a lo más alto gracias al talento español. Conoce ECUSA. Únete a ECUSA.
Then I'm going to invite Antonio to introduce our speaker, and we can start with the scientific part of the event. Thank you for listening.
Hello everyone. Welcome and thank you for coming to our ECUSA California event. Today, it is my pleasure to introduce our keynote speaker, Dr. Eva Nogales, a Howard Hughes Medical Institute investigator and professor of biochemistry and molecular biology at the University of California, Berkeley. Dr. Nogales started her path like many of us. She obtained her bachelor's degree in physics at the Universidad Autónoma de Madrid. She then moved to do her graduate work at the synchrotron radiation source in the UK, where she used cryo-electron microscopy to investigate the assembly of tubulin polymers. She then did her postdoc work in the lab of Ken Downing at the Lawrence Berkeley National Lab, producing the first atomic structure of tubulin using electron crystallography. Finally, she joined the molecular and cell biology faculty at UC Berkeley in 1998. Currently, Dr. Eva Nogales holds the position of Senior Faculty Scientist at the Lawrence Berkeley National Lab and a Professor of Biochemistry, Biophysics, and Structural Biology at UC Berkeley, and she has been a Howard Hughes Medical Institute investigator since the 2000s. Dr. Nogales' lab is dedicated to gaining mechanistic insight into large macromolecular assemblies that could potentially illuminate biological functions by direct visualization of their architecture and regulatory interactions. The Nogales lab uses state-of-the-art techniques such as cryo-EM and image analysis, as well as biochemical and biophysical assays. She has also received many awards, and countless. Among them, we would like to highlight the Dorothy Hodgkin Awards from the Protein Society, the Portrait Lecture Awards from the American Society of Cell Biology, the Biophysics Lecture Award from the Biophysical Society, and the Greenway Medal from the University of Melbourne. She is a fellow of the American Association of Science, the Biophysical Society, and the American Society of Cell Biology, where she served as president in 2020. She is a member of the USA National Academy of Sciences, a foreign member of the Real Academia de Ciencias de España, and a foreign associate of the European Molecular Biology Organization. And the list goes on and on. Thank you, Dr. Nogales, for accepting our invitation to be the keynote speaker at this event. It is our honor to have you here. Despite getting countless awards and doing wonderful and cutting-edge research, Taking your time to give a talk at our organization event is a true example of a leader. We will never forget the day that we were nervous to invite you to be here today. And just 1 hour after sending our email, we got the most humble response of, "Yes, I would love to participate." This truly speaks of your character and your passion of giving back to the scientific and space community. Thank you for being here today to share your time and expertise with us. And thank you for being a true example of a leader that inspires us, Americans or Spanish, women or men, to advance the wonderful world of science and be leaders of the future. Please, everyone, join me in welcoming our keynote speaker, Dr. Eva Nogales. Thank you.
That was so sweet. Thank you so much. I have to say that I was watching and listening to the video, and I was thinking, where was ECUSA when I was a postdoc in Berkeley, and I felt so lonely, and every time I encountered a Spaniard, it was like, wow, and then, I became faculty, and everybody that I met were typically graduate students of postdocs. They will come, we will become very, very good friends, and then they will leave, and every time, it was like a little piece of my heart went away with them. They still visit, I visit them, but I think it's a fantastic job that you guys are doing, and I congratulate you on your accomplishments, how many people are part of this, and the vision that you have is fantastic. And thank you so much for inviting me, be part of you want me to take it off? Or?
I mean, you want. The presenter can remove the mask.
Can you hear me with it on?
Yeah.
Is it okay? All right. All right. So what I would like to do today is to give you a glimpse of the kind of research that we do in my lab. I will illustrate it with a couple of examples. I prepare lots of slides but I don't have to go through them. If we make it through half of them, that would be fantastic, 3/4, whatever. I would rather you interrupt me with questions. This is such a cozy, I don't see you because I'm completely blinded, but I know that you're there. Please interrupt me at any point. I am completely in teacher's mode. I'm teaching at Berkeley right now. My final exam is on Monday. I think I'm gonna ask questions and test you at the end of this because I'm in that mode. Anyway, so I would appreciate as I go, I will build it. If there are people that do biochemistry or biophysics in the audience, they will get everything if very easily. Others, I hope that I build it slowly enough that if you ask me questions as we go along, you will get something out of it. And at least like my mom saw me once presented at a conference in Spain, and she's not a scientist, she didn't even go to high school, but she told me at the end that she loved it because there were all these pretty colors. So I hope you like the colors of what I have to show you. All right, so again, for the non-biologists in the audience, and sorry if you're all biologists, but let's start from the basics, which is kind of a fundamental principle. It's so fundamental that we call it dogma, but dogma has a bad connotation, but it really means the absolute basics that form the molecular foundation for what we call life. And it has to do with this passage of information from the DNA, which is our genome, to the units of function in the cell, which are proteins, through this intermediate, which is RNA. So these are different types of biological polymers that work in this flow of information in this dogma of molecular biology, in which everything is funded from the cellular to the organismal level. So the unit in DNA that contains information that will lead to a protein is what we call a gene. And there's a fantastic molecular machine in the cell called RNA polymerase that copies this gene into a special type of RNA, which is a messenger RNA. And then there is an even maybe more fantastic molecular machine, which is called the ribosome, that is able to translate this change of nucleotides into a change of peptides, a polypeptide, a protein. And these proteins are the functional units in the cell, and I'll show you some some examples that we have studied in my own lab. So, I will define myself as structural biologist, which means that I'm interested in defining, characterizing, and finding meaning in the structure of these biological macromolecules that I just told you about. And what is the point of defining the structure of these molecules? One has to do with the search for a fundamental understanding of what are the molecular bases of cellular function. We want to understand how these proteins, this RNA, this DNA work at the chemistry and physics level. Chemistry, contact, physics, motions, forces. And you know, at the end of the day, we're generating ever larger and larger amount of structural information, visual information. I love these images. They are created by David Goodsell, who is both a scientist and a fantastic artist that bases all of these drawings in real structures and real information about concentration and where they are located, one with respect to the other. So hopefully my lab and that of other structural biologists contribute to generating these kind of galleries of how all these molecules are shaped and how they interact with one another. There are obviously also practical applications, and these have to do with drug design, and I'll show you a couple of examples as we go along. The idea is that the shape and the motions and interaction of a protein define their activity, and that if we know about it, we can, in principle, modify it, we can enhance it, or in most cases, because it's the easiest, we can eliminate it. So this image here is just a part of a protein that I study that is called tubulin. It's a little pocket that binds this purple molecule that is Taxol. And Taxol is the most broadly used anti-cancer drug for the treatment of solid tumors. And it acts by binding to this particular pocket and then having an effect on how tubulin works. And I'm going to tell you about that later on. But is it easy to get the structure of this molecule? So let's think a scale for a second. So the things that we deal with on a daily basis are of the size approximately of a meter in that kind of regime. So a puppy, a table, a car, they are all fractions of a meter or so. Now, these are things that of course we can see with our eyes. If we look at a flea in the leg of the puppy, that's 1,000 times smaller. And for that, many people can see it. I already cannot see it very well. I will need some magnifying glass. But if we look at a bacteria that is in the leg of the flea, that is in the leg of the dog, this is now the size of a micron. And that's a millionth the size of, a millionth of a meter. And for that, we will really want to use a powerful optical microscope, at least to see the presence of the bacteria. But this is not what I'm talking about. I'm talking about seeing the molecules of which there are millions inside of a bacteria in atomic detail. And now we're talking in the scale of a nanometer, which is a billionth of a meter. And for that, light is not good enough. And we need to use electrons, and this is why we do electron microscopy. Now, it's very interesting because the smaller the thing you need to look at, the larger the microscope gets. So this is just an example of an electron microscope with me there for a scale. Normally, many of these microscopes need special rooms with high ceilings because otherwise they don't fit in. In any case, these electron microscopes really have the power of giving us images with very high resolution. They're used not just in molecular biology, they're used even more so in physics and in material sciences. And what you see here is an image that has a resolution of less than 1 angstrom, which is 1/10 of a nanometer, which I was telling you was a billionth of a meter. So this is such that individual atoms can be seen in a study like this, which is a film of silicon for which electron microscopes are used a lot. Now, this is an inorganic material where you can put thousands of electrons going through each one of these atoms to get a really contrasty image. But we cannot do that with biological material. So we are really limited, and we have to overcome many obstacles. Why? Because biological molecules, first of all, they hate the vacuum. An electron microscope operates under ultra-high vacuum. The column where the electrons move and where the sample has to be introduced is under very high vacuum. They also are made of very light atoms, carbon, nitrogen, oxygen. So they have very, very little contrast with respect to the background of water, which is where they like to live on, not in vacuum, but in an aqueous solution. But ultimately, the thing that is really limiting is that they are radiation sensitive. You pass electrons, so highly energized electrons, like the ones that we use that are moving at close to relativistic speeds, and they fry the sample. So we cannot put that amount of electrons, like I showed you in the image before, to study them. Now there is a solution, and the solution is called cryo-electron microscopy. So the cryo in the EM is what is special for the study of biological molecules. And this involves a process that is referred to as vitrification, which is a process of very, very fast freezing at rates that are of the order of hundreds of thousands of degrees per second rates, okay? And this allows the freezing that is so fast that the water molecules don't even have time to move into the position of a crystal. So they don't make ice. They make a solid, amorphous form of water that really preserve the hydration layer and the way proteins interact with water and keep them in very good state. It is possible of high resolution with some caveats, which is contrast remains low. So what does the cryo mean? Cryo means that now this solid water can be introduced, if it maintains at very low temperatures, liquid nitrogen temperatures, can be introduced into the high vacuum of the electron microscope without any sublimation of the water. So the water remains, and the particles, the molecules are perfectly happy in it. The second is that it reduces, it does not eliminate, but it reduces radiation damage. This is because when electrons go through the sample, they kick electrons off your sample, of the atoms in your sample off, generating ions, radicals that then move in the water and cause a cascade of reactions that break every covalent bond apart and make your protein, your DNA, RNA, really destroy them, okay? So by keeping the sample at very low temperature, although that initial radiation happens, the radicals don't have much opportunity to move because of the very low temperatures. So the diffusion of those radicals is reduced, and radiation is reduced with it. However, it is not eliminated, so we have to use very low doses, typically 100 times less dose than material scientists will use for their images, maybe 1,000 times less. So we use what they're called low-dose techniques, and we deal with very, very noisy images. And after we take those images, we use computation to deal with that low signal to noise. So let me show you the pipeline of how that happens. So the first thing that we do is we purify whatever it is that we want to study out of the cell using biochemical techniques. And then we have, say, our protein of interest in a solution where it's happy and doing its reaction. And in there, molecules are just diffusing in the water and being able to catalyze reactions, self-assemble, whatever it is that they do for a living. We take a small drop and we put it on an EM grid. And this is an EM grid, a copper one, that you see for a scale with the thumb of Gabe Landa, who made these images. Then we put this in what we call a vitrobot. It's like a robot for the vitrification process that has a chamber where we can control things like temperature and humidity. And then we do something brutal, which is thin as that little droplet of buffer with your protein is in the EM grid, is still too thick. And we have to make it very, very thin, because otherwise the electrons interact so many times that with your with proteins that at the end you don't see anything. It has to be a layer that only contains 1 or 2 proteins, so just a few nanometer thickness. And to do that, we come with filter paper over the EM grid, and then we quickly dip it in something like liquid ethane, something with a very high heat capacity that allows the very quick vitrification of the sample. And in doing that, we go from these molecules in solution to a situation in which we have just a thin layer of vitrified water with the protein in it. And this is what is going to go inside the vacuum of the electron microscope for imaging. So there it is, our protein of interest, and we're going to shoot electrons through it, and the images will look like that. The images are projections. I could tell you more about why they are projections and not a section or a surface, but the whole point is they contain information on the whole structure. It's like the addition of the density in the direction of the electron beam. An equivalent from our daily lives is an X-ray when you go to the doctors. An X-ray is also a projection. It's not the tip of your nose or the back of your head. It's everything collapsed into 2 dimensions, but that contains information of the whole thickness of your body. So this is an actual real image. This is how things look like. And this has many copies of the molecule of interest. We actually select them to generate a gallery. We used to do this manually, and we'll pass it on to the undergraduates. Now we have artificial intelligence that for us. Then we classify, we get all the images that are in one view, and then we align them in plane. And that alignment, which is maybe gonna, that alignment then allows us to add them up, sum them up in the process of average that really boosts the signal. So now the features become more obvious because that background that doesn't add in a coherent way disappears while the features in the object do add up. Okay, so this is the way we overcome the problem of that very low contrast that we have because we're using very few electrons, taking images with very little light. But that only gives us a 2-dimensional projection. We have to do that for all the orientations. We have to computationally find the relative orientations between them and then put them together to generate a 3-dimensional density. And that 3-dimensional density is what we now are going to interpret in terms of the polypeptide chain that follows a certain path that gives shapes to the protein. Now, in many cases, I'll show you examples. What we study is not in a static conformation. It's moving. This is part of how these molecules work. And it means that when we go through this analysis, we can actually typically obtain more than one structure. We study as many structures as they are present in solution for our biochemical conditions. Okay, so this video was generated quite a few years ago when this was the state-of-the-art resolution, which is just better than 10 angstrom, okay? So about 6 or 7 angstrom. The whole point is that these density maps can be interpreted in terms of that path. What you see here in these colors is what is called a ribbon diagram that just schematizes the path of a protein, where in this section is what is called an alpha helix, and this is a loop and so on. When the resolution gets to be better than 4 angstrom, we start seeing specific features of individual amino acids, and we can do what we call the tracing of the polypeptide chain ab initio from scratch. Okay, so those are the principles. Questions? Yes?
How do those structures compare to computationally generated?
Right. How could I not get a question about AlphaFold and Rosetta Fold and all that? Yeah, so if Rosetta Fold gets it right, it compares well. I will tell you that the kind of thing that we study benefit tremendously from having an initial model that may not be perfect, but we can use as a starting point to then refine and see how things are in the context typically of larger complexes or polymerized structures. So this is more involved because you have to purify the protein, do the imaging, the computation, but what you get is conformational states, larger assemblies, different states with different bound ligands, and things like that. Yeah.
Is the refinement is usually done through molecular dynamics, right?
No. Molecular dynamics is what you would do, say, when you have a structure and you want to interpolate motions that will happen under a certain buffer temperature conditions that last a few seconds. So that's based on physical principles. But what you may be thinking about is that once we have a density, we can use for the refinement, the improvement of initial model that we do, the tracing, we can use all the methods that X-ray crystallographers have used over the years. Although in our case, the density map is the real density map. While in their case, it's it's a kind of extrapolation that it gets improved in an iterative fashion as they build the model. But otherwise, yeah, we can use all the tools that have been built in the X-ray crystallography community over the years for that. Then molecular dynamics can be used on any structure, whether it's generated by alpha-fold crystallography, cryo, to do interpolations in time of how things move. So those, yeah, that's a kind of advanced question. Any other things? Yes?
Many proteins have different structures based on physical conditions, ligands available, and so on and so forth. Is there a way to account for altering structures while this technology?
Sorry?
Is there a way to account and take into account these altering, formations and structures?
Right. So one of the things that we can do is there are proteins that change conformation. I'll show you an example upon binding to a small molecule in the cell that controls their conformation. And we can put them in the presence of different cofactors, small molecules, drugs, whatever, and see how they're changing. In many cases, they change upon interaction with each other. We can reproduce all of those conditions based on some previous knowledge that tells us that's going to be interesting, and then see what is happening at the structural level. So this is just a summary. This is the sample that is embedded in this thin layer vitrified water. Electrons go through it, and we generate images that can be averaged and then combined to give us a three-dimensional structure. The thing that is important, I was talking just now about the fact that X-ray crystallography is a method that has been used to obtain protein structures for decades, and is still the one generating the most number of structures that are being deposited in the protein data bank. Maybe not the most exciting, because the most exciting are being generated by cryo-EM, but there are still many. Now, crystallography really is a very powerful technique to obtain really high resolution structures, but it requires that you get a lot of your protein, and that you find conditions to crystallize it, that the crystals are ordered enough, and that then from the diffraction data, which is not real space data, you deduce what the density is, which is called the phase problem. So there are a number of bottlenecks, and as the protein or protein assembly gets larger, or if it has a special conditions, like it's an integral membrane protein, and it has to be in detergent, it just becomes more and more, and it's harder to overexpress, becomes harder and harder to crystallize. So the whole value of this is that you do not have to crystallize. Yes?
And to sound weird, Ada Jonas got the Nobel Prize for the structure of ribosome.
Yeah, that's a ribosome, by the way. That's a ribosome.
The question is, I understood that it was very hard for her to get the crystals.
To phase it, the crystals were easy. What was difficult was to phase it and to even collect the data, because I can go on and on about this. Because I lived in synchrotrons during my PhD, and my husband is the head of the experimental system group at the Advanced Light Source, which is the synchrotron in Berkeley, the reason why we came to California. So ribosomes are huge, which means that in reciprocal space, the diffraction pattern, the spots are very close together. You need, special beamlines, special detectors to be able to separate them to get good data. But then the most difficult thing is to face it. How could you face something like that? And it required developing of a special molecules of heavy atoms, because individual atoms will not do it, that will bind to be able to have the facing power for this process to occur. And eventually occurred by Ada and by others, but it was very tricky. Now, no one bothers to crystallize ribosomes. And the highest structure of ribosomes have been obtained by cryo, and they is the ribosome with translation factors, initiation factors, termination factors, recycling factors, translocation into the ER, the whole thing, because you don't need to crystallize, you don't need to solve this, So this makes a big difference. So no crystallization, fully assembled complex, so ribosome with many other things on top of it. And for ribosomes, the reason why it wasn't difficult to crystallize ribosomes is that if you look inside a cell with an electron microscope, everything in the cytosol is ribosome. You can purify it by the bucket. The things that we study, transcription initiation factors, they are minute amounts. It's just not possible to simply purify them, and they're too large to do very efficient reconstitution. When you have to rely on endogenous material, you get very, very little. And right now, it doesn't matter. You can use CRISPR to tag the endogenous subunit of one of these complexes with a tag that you can use in purification biochemically. You take it out of the cell in whichever amount you can do cryo. So this has been truly revolutionizing. Now, cryo has been around for a long time, because I was doing it for my PhD, and I'm really old. Fall out of my hair. But something happened just a few years ago when we were really limited by the detector technology. So all of these, at the end, you have to record the data somewhere. And for a while, I was going into the dark room at the end of the day, turning the red light on with film, developer, fixer, scanning afterwards. And then there were detectors, there were CCD cameras that were terrible, absolutely terrible. And about 10 years ago, new detector technology was developed and was commercialized, and it changed the game. So these detectors, I will go on for too long, so maybe with the paella, for those that are interested. But they have made a difference of at least 2 orders of magnitude on what is possible in all directions. You can study almost anything. You can get to super high resolution. If there are multiple structures, you can separate them. It's been truly amazing. So now it is typical to get a number of states that describe the equilibrium of whatever number of components that you have in your mix. And it is possible to get to very high resolution. So typically the resolution are in this regime, but now it has been demonstrated you can get to 1.2 angstrom resolution. That is using the absolutely top of the pops, microscopes and aberration correctors and detectors and all the rest, with the best behaved samples. The ones that you use to demonstrate that the methodology is possible. But it tells us what is theoretically, not theoretically, it tells us what is now physically possible with the technology that we have. So the whole thing is, you can look at anything. It's really quite amazing. So I've seen it growing and expanding, evolving, and then, slowly evolving, and then taking this really step up to where it really has become quite an amazing technique. So these are the 3 guys that ended up getting the Nobel Prize for this. Jacques Dubochet was the one that developed the methodology for vitrification. So until then, the person that didn't get it, I have to say this because, was Robert Glaser, who is from my university. He was the first one that demonstrated that if you freeze the sample, you can obtain the information that you require for high resolution through this reduction in radiation damage. But it was very hard to reproduce the conditions that he got for freezing the samples, and Jacques Dubochet was the one that developed that technique so that now anybody can do it in their lab. Richard Henderson was the first one, he's Swiss. Richard Henderson, who's British, was the first one to use electron microscopy to get an atomic structure. That was the one of bacteriorhodopsin, using something that is not exactly the methodology that I told you, is electron crystallography, which uses two-dimensional crystals. But in any case, he was also the visionary that said, this is theoretically possible, and the most important limitation is the detector, and he was one of the people working in the two teams that developed this technology. One was here at the Lawrence Berkeley National Lab, the other one was in Cambridge, UK. And then the last one is Joachim Frank. He's German-American, and he was the one that developed all these computational pipeline that I told you, the classification, the alignment, the three-dimensional structure, all of that was developed by him. They did this in the late 80s, beginning of the 90s, but it was only in 2017 that they got the Nobel Prize, after the direct detectors, these new detectors, made the whole technique explode in usage and resolution, at which point the Nobel Committee realized the importance of the methodology. So let me show you some examples. I just told you that in this central dogma, there is a process by which a gene is copied into a messenger RNA that has a name. It's called transcription, and is carried out by the RNA polymerase. Even viruses have their polymerases, and of course you would have heard about the RNA-dependent RNA polymerase of SARS-CoV-2, which actually is the one piece of the whole virus for which there are small molecules developed as antivirals. So everybody has one of a polymerase like that. That one in that virus is not the simplest. There are others that are even simpler. Bacteria, it gets more complicated. Eukaryotes, it gets really complicated. And that's what we study. We study the most difficult eukaryote, humans. This has a double meaning. In any case, one of the things that has to happen in all cases and different life forms use different strategy is that out of the sea of your entire genome, the polymerase has to find the beginning of a gene so that it starts transcribing to where the gene starts, and not before, not after. And it has to do it accurately, because if it gets out of frame, things are going to go really screwed up. So how does the start of a gene is found? And the polymerase doesn't know how to do that. It needs other factors. And in particular, it needs this factor, transcription factor 2D. The D has to do with the order in which these things were discovered. This is a complex of proteins. It's 20 proteins. It adds up to 1.3 megadaltons. And this has the capacity to read special sequences in the DNA that are around the transcription start site. And then it recruits the rest of the machinery, which includes the RNA polymerase itself, which is also a large complex, 12 subunit, half a megadalton, other factors, and also TF2H, which I want to also mention, because it's also very large, and this is a real machine that uses energy in the form of ATP hydrolysis to actually do something very important. So everybody, even if you're not a structural biologist, even if you're not a molecular biologist, even if you don't know about science, they know about the double helix of DNA. There's a beautiful structure, it's the beginning of a structural biology. It tells you so much about how DNA stores information and can copy it and repair it and things like that. Very stable structure. But it has to be open for the polymerase to be able to read one strand. If the two strands are paired against each other, they cannot do that. So energy has to be used to open this very stable storage form so that the polymerase can get in. That's what this subunit does. This large complex does. So we've studied all of this. I just want to show you, and this was way before the direct detector, so where we didn't have very high resolution, that the complex, which is huge, compared to a regular average protein, will be like will be like a dot in here, okay? Most of the proteins that are done by extracrystallography. Just the internal joke. Okay, so here I hope you can see there is something, there is a lobe that is moving across. We can quantitate this. We can see how this yellow piece moves from one side to the other. It occupies two different positions and it moves across them. And very interestingly, how many of these molecules we see in one state or the other depends on whether we give it DNA, which is what it likes to bind to. And it is because one of these states is the one that can bind the DNA, and the other one is completely inhibited. So this molecule has an inherent flexibility between two states, one of which is functionally important for DNA binding, and one of which is completely inhibited. And it exists in this equilibrium. And this is fabulous because one of the roles of this complex is that it will bind to the start of a gene only when that gene needs to be expressed. So it has to communicate with other factors in the cell that are doing the reading and the signaling to tell it is time now to bind here. So it has to have the flexibility to be able to switch between an inhibited and an activated form. Okay, yes?
Well, maybe very basic, but how do you manage to purify this?
Okay, very good. So this particular one we purify by immunopurification. So there is an antibody that recognizes an epitope in one of the subunits. You bind this epitope to a column, pass nuclear extract from huge amounts of HeLa cell. The complex bind, and then you elute it by using the peptide, a synthesized peptide that competes, and it comes off. And that's it. That's all we do.
And how come it doesn't fall apart?
It doesn't fall apart because this is a stable complex. There are many complexes in the cell that, as a part, that form this only transiently. I will tell you. I will show you in a second how we study ones that are transient. We mix them together. They exist in a mixture, and then computationally we separate the ones that are fully formed from the ones that are not. But in this case, you pull out of it, you grab it in the cell, pull it out, and it comes as a stable complex. The same for the polymerase, the same for TF280. That's what we call them complex. They are constitutively of enough affinity that they don't come apart. Yeah.
How many projections do you need to make for a movie like this?
Yeah. So that little movie was just class averages where we align the part that is fixed, that is larger, and then we see how the other one moves. It's just a superposition. It's not a real movie. I think it's very important that you ask that question. We don't have movies because everything was frozen. It's just that we froze them where each molecule was in a different state. So we can now put them all together and see the whole motion. What we have lost is the speed. Obviously, we can't get information on the speed. To get information on the speed, we have to use other techniques that are real time. But we get all this information that has to do with, all of the states exist. Every time we get a structure, we have to split the data into the part that go to one state and to the other. When you have a continuum, you can subdivide ad infinitum, but you end up with no data. So you make some, compromise on how much the data is split. But what requires a certain amount of images is to have a structure at a certain resolution. So typically, the number of images that go into one of these structures is of the order of 20, 50,000, 100,000 images of the object. Typically, one micrograph may have 100 or 200. And we take thousands of them. So we start with 1 million, but some of them we just discriminate because, again, they're in some transition state that we cannot. So ultimately, in terms of signal, we are averaging of the order of tens of thousands of images of the object. The computation is tremendous. It's a lot more than X-ray crystallography, but not as much as AlphaFold. So it's kind of in between. Yeah. So we've studied the process of TF2D now at high resolution, how it moves in the absence of DNA, how it engages DNA, how it recruits the polymerase, how it recruits TF2H, how TF2H opens the DNA. I'm going to summarize it into an animation that we made with Janet Iwasa, who's an expert on this. Okay, so this is it. This has just some names. The thing that is very important is that red protein, which is the one that needs to bind the DNA and recruit other factors, and which is kept inhibited, completely inhibited from binding anything. TF2D comes to a certain site by recognizing certain histone, blah, blah, blah. But binding, recognizing certain sequences, allows the red factor to start searching for its binding site, and that is facilitated by TF2A. And now these have served as a kind of, sorry, I'm pointing at my screen. Sorry, let's do that again, because I'm not going to tell you about microtubules, I don't think. I'm not going to have time. So that piece, what it is released, brings in the polymerase, which comes with another factor, TF2F. And now this is the platform for the binding of the next one. Each one of these steps we visualize. And then this recruits TF2H, which is in one structure by itself. And then as upon recruitment, you will see changes conformation. It kind of opens up. It's again in an inactive state until it engages with the polymerase, opens up, and that blue part is the motor. It uses ATP to walk on the DNA, pushing the DNA towards the polymerase and unraveling, so that now the individual strand can go into the active site of the polymerase and it can start copying it. Okay, so the one thing that I want to show you is that we have got many of these structures. One of the ones that we got at highest resolution is a dangling part of that TF2H that has another function. It's a kinase. It has to do some modification of the polymerase that is important for the polymerase to start moving into the gene. This kinase functions regulating transcription, but it also regulates cell cycle and is a major target as an anti-cancer. And we have looked at one inhibitor that is in phase 2 trials, and we've done cryo-electron microscopy. We have, in this case, a structure at 2.5 angstrom. We can see the molecule. We can see waters that are involved in binding to the molecule. And this is the kind of information that is needed to understand how this inhibitor is specific for this kinase and is not going to have secondary effects. How could it be modified to increase its affinity and its potency? Okay, so I'm showing you this because I started at the beginning saying we want a fundamental understanding, and we've learned a lot about how transcription is initiated. But also, I told you, this is important for drug development and drug characterization. So I have another whole lecture to give, but let's leave that for Monday. I just want to tell you that this is something that now, with this kind of resolution and this kind of applicability, it has moved from a world that was completely academic to the world of pharma and biotech. I'm thinking genetic, because Genentech now has bought the most amazing microscopes, and they have made a huge investment on it. And myself, I'm a co-founder of a company that is set in Boston called MoMA Therapeutics, for molecular machines, not for modern art museum. And we are using cryo-EM. Now every time we have a lead compound, we can look at it bound not to a domain, but the whole structure of these large molecular machines to understand how they work. And in some cases, it is wedging between the protein and the DNA, and this is something that it would have been much harder to visualize by crystallography. In any case, what I want to do is just thank the people that were involved in this work. Because even with that quick movie, I went through a lot of work that was for TF2D initiated by Patricia when she was a postdoc in my lab, and she decided to join my lab, and she's been a senior research scientist in my lab for now many years. Mike was the one that characterized that motion of TF2D and saw that it relates to the binding of DNA. Now he has his own lab at University of Michigan. He was a biophysics graduate student. And then two other biophysical students that are now doing their postdoc, Robert and Abby, were the one that ended up using the new detectors to get the structure at high resolution of TF2D by itself and bound to DNA. Yuan was the one that did the work on the assembly of the recruitment of the polymerase, the recruitment of TF2H, and that very high resolution structure, and the structure the whole TF2H out of the context of this transcription complex, but how it exists in solution, and the high resolution structure with this inhibitor is the work of Basel, and they both have their own lab, one at Northwestern University, the other one at the Institute for Cancer Research in London. And then Jay is my lab manager. She purifies these complexes from huge amounts of HeLa cells, and Dan also runs the cryo-EM facility at UC Berkeley, which is a state-of-the-art facility. And with this, I think I'll put it out. That's about a third of the slides that I had prepared. All right. I don't know if there are any remaining questions. Yes.
Just so I'm sure I understand, how do you go from the view movie of just the complex to the whole movie?
Right.
Do you actually see all this, or it's an assumption?
No, it is an interpolation between states. We have TF2D in several states, so we see how it moves. We see how it binds to the DNA. We see it bound to the DNA. We then see how.
So you've done primary among
so
TF
that
plus the complex.
Yeah. So that comes out of about, I would say, 10 to 12 different structures, snapshots, and what you see in between is just an interpolation, right? Where you have one structure, you have the next, and between them something came and bound. So that is the animation part of the. This is not molecular dynamics. There's no computer in the world that could do that, right? Molecular dynamics. So about 12 different structures, built at different states, different conformations, and things like that, just put all together.
So you cannot get all those complexes, the whole pole 2 complex with all the.
Yeah, there is a structure of the whole thing put together, and even bigger, because there's another factor called mediator that has also been seen bound on the other side of the polymerase. That's not our work. That's why I haven't mentioned it. That's even larger.
But it's done separately and then put together. That's what I mean.
Sorry?
It's done separately and then put together.
No. This is the whole thing. Yeah. It's not one piece or the other, and then I just think that they're going to click like that. No. The whole thing. We can do them separately, and then we combine them biochemically, we image them, and we see the whole complex. So this is like, it's 3 megadaltons or something like that. Yeah.
When you say that you combine it biochemically, you mean you are...
We're mixing them. You purify the polymerase, the TF2H, because those are stable complexes. And then we put them together on the DNA that has a transcription start size that is being bound, recognized, and the whole thing builds in it. In the test tube. And there's an equilibrium because there's complexes that are coming and going. But we study them and we see the ones that are the full complex, the ones that are intermediates, and so on.
Okay, thank you.
Yeah. Yes.
Is it possible to also do this for transmembrane proteins, perhaps in liposomes?
Absolutely. So this has been. Now, the number of structures of integral membrane proteins is growing exponentially because, again, you don't need to purify these in very large amounts, which is very hard for integral membrane proteins. You have to solubilize them with detergent to get them out of the membrane. And in EM, you can also reconstitute it into nanodiscs and other things so that you have them in an environment that is more like what they have in the cell. And there are zillions. I mean, the number of ion channels, transporting, photosynthetic systems, channels of all sorts is exploding. Yeah. Many of these integral membrane proteins are major drug targeting for many different types of diseases. So it is really, I think that's what really got the attention of the pharmaceutical world. Yeah. This side is winning. Yeah.
I have two questions. The first one is when you were showing us the distances between the atoms of the molecule, are those lengths studied by the intensity of the ray?
Yeah. So when you are at 1.2 angstrom resolution and you see individual atoms, including hydrogens, you can deduce that directly from the map. We don't, and even most crystallographic studies, they are built on tables. So when we do the refining, we make sure that all the bond length, bond angles fulfill, physical criteria that we know are what they have to be. That's part of the process of building the polypeptide chain that allows us to do it with high accuracy, even when we don't see every hydrogen atom. But we can model it because all that information pre-exists, and it goes into the building of all the models in crystallography and in primary.
Yeah. The second one, yeah, I said.
The second one?
Sorry. The second one is how long is the time taken to that movement?
Right. The motion that I was describing, we have no idea. We have no idea. So we are doing now the experiments using something, a technique that can give us real-time information. So what we are planning on doing, and we are already designing and doing the initial test, is what is called single molecule FRET, fluorescence resonance energy transfer. Thank you. So this is a method by which we could put some label that is going to be able to read out the distance between the two domains in places where we know are going to be sensitive to that motion. And that data is fluorescent data that can be collected in real time. So that kind of information that has to do with rates of motion have to be obtained by other techniques that are real time. Maybe they don't give you structure, but they can give you distance and do that in real time. So you can interpret that data based on your structure. You can both design the experiment based on your structure and then interpret the data based on your structure. But that the kinetics of the process have to come from something that is a kinetic technique.
And do you think could it be done instead of this electron microscopy that you have to frozen the samples so you cannot see the movement
right.
you are working with it? So could it be used by, for example, scanning tunneling microscopy or atomic force?
Right. So atomic force microscopy will be for biological material. So those do not have the kind of resolution that we want, and the time resolution may not be sufficient to see this kind of thing, right? So yeah, so I think that can be used for things of other scale and with other kind of time courses. This is going to be a little faster for them and a little too large for everything. There was a question from this side. Yeah.
So what is the size limit of the lower size limit that you can image?
Yeah. So the smallest thing that has been imaged today by EM, I think is of the order of 50 kilodaltons. The idea is that in principle, with improved optics and something that is called a faceplate, I could go for an hour on that one only, that could be reduced to the limit of 30 kilodaltons. But to be honest, what is the point? Because something that is 30 kilodaltons, if it has a well-defined structure, AlphaFold is going to give it to you in seconds.
Yeah,
so what is the point?
Membrane proteins, for example, that are very
yeah,
small.
AlphaFold is getting better and better. It's true. So for those, so there's people that are really pushing the technique to deal with very small integral membrane proteins, which are difficult by NMR, which is the 1/3 technique that I haven't mentioned. They are difficult to crystallize because they're integral membrane proteins. But AlphaFold has a little bit of a harder time because at the end it needs data to be able to build. And right now we don't have as many integral membrane proteins as we have soluble proteins. But that's the limit. On the upper limit, there's no limit.
Yeah.
So yeah, I think that if you have something that is 30 kilodaltons or 20 kilodaltons, unless it's an integral membrane protein, you will do better just trying to crystallize it or just at that point using alcohol. Yeah. Yeah.
This is beyond my expertise, but it looks like you have several ways to relate to an structure. So how do they compare if you look at something obtained by cryo crystallography or AlphaFold? I think you say
AlphaFold, yeah.
AlphaFold. I mean, when you have something, a benchmark, how do you look, when you compare them? Do they look that identical?
So there are many examples now of cryo-EM and crystallography of the structures of the same thing obtained at the same resolution. They look practically identical. There may be differences that have to do with how things that are disordered are visualized, how much or how little each one of them is able to tell you about things that are kind of flexible, within the structure. But they look basically identical. The one thing that happens is that this is a little different, but at the kind of doses that we use, there's still a little damage that occurs on negatively charged residues. Aspartates and glutamates tend to get this decarboxylated. So you may not see some of the densities for the ends of the side chains of those, but otherwise they're the same. So AlphaFold, it just depends, because if it's a protein that then forms part of a complex and it has been obtained by itself and it undergoes some conformational changes when it binds to something else, what you see doesn't necessarily correspond. Now, RosettaFold, in the last issue of Science, if you saw this is David Baker's, is starting to look at complexes. I mean, honestly, it's amazing. So they're doing pairs, and even there are, in a few cases, they've been able to do 3 proteins at a time. So they're getting better even in the context of complexes. Still, not the kind of thing that we can do by cryo-EM, but they're getting close and closer. Now, it is very hard for them to be able to predict conformational changes or effects of small molecules and things like that. That still will require experimental techniques that really are down to the details, but that they can use the AlphaFold as a starting point. AlphaFold is really going to help us structural biologists if they're looking at the right things. If they're looking at the wrong things, they're going to be debunked by AlphaFold. They're going to be redundant. But if they're looking at the right things, another level of complexity, AlphaFold just gives them a step ahead towards the final end.
So thank you, Dr. Nogales, for a wonderful talk, and thank you everybody for all your questions. So we would like to give you a little present from our association for thank you your time and your expertise with us.
I hope it's Spanish wine. Yeah, thank you so much.
Now I would love to give the word to Doina, which is going to introduce the next part of this event, which is a roundtable discussion. So, yep, all yours.